Professor Anna Nekaris. Anna's main focus is on the conservation of Asian nocturnal animals, especially slow and slender loris. Her primate conservation work extends to African nocturnal primates, lemurs, columbines, and macaques. She's published over 250 peer-reviewed articles, and with nearly 30 years of research in 11 loris range countries, Anna is basically a leading expert in Asia's slow and slender lorises. She's also director of the little Fireface Project, which I'm sure she'll talk about throughout her talk, as well as afterwards as well. She's the member of the IUCN Primate Specialist Group and the co-editor-in-chief of Folia Primatologica. I hope I've said that right. So I'm going to pass you over to Anna, who's going to talk about the ultrasonic communication to maintain social cohesion in the territorial and venomous Javan slow loris. So over to you, Anna.
Hello, my name is Professor Anna Nekaris, and thank you so much for inviting me to give this talk today. I am a professor at Oxford Brookes University in primate conservation, and I'm also the director of the Little Fireface Project, which is a conservation charity working to conserve slender and slow lorises in Asia, particularly in West Java, Indonesia, where I've been running for 10 years, a long-term continuous behavioral ecology field study of the Javan slow loris, which you see here. The topic of today's talk is to describe to you their ultrasonic communication, which is something we discovered during the course of these 10 years that we now know helps to maintain social cohesion in this relatively gregarious nocturnal species. Slow lorises are all globally threatened. They are all critically endangered, vulnerable, or endangered on the IUCN Red List. I am working on the Javan slow loris, which you see in upper left. It is critically endangered because of illegal wildlife trade, black magic, traditional medicines, and of course, habitat loss, which is something that all of these slow loris species face. Their biology is relatively unique. They are one of the only exudativorous primates, eating gums that they can make flow from trees through specialized teeth that gouge holes in trees. This is more than half the diet of most species. They also consume a lot of flower nectar, and they are important pollinators of these flowers. And they also consume a lot of insects, including incredibly toxic insects. You will note that they very rarely eat fruit, despite living in tropical forests and plantations, such as agroforests, which I'm going to tell you about today. They even live in mangrove forests where they are known to swim, so they're an incredibly adaptable species. They occur all the way from Northeast India to the Philippines, and the smallest species is this one, the Philippine slow loris, with this one, the Bengal slow loris, being the largest. And you can see they have an incredibly variable body size, from 350 grams all the way up to 2 kilos. If we look at some unique aspects of their biology, they live in small social groups. They tend to be unimale, unifemale, with a single male and a single female, which we know can be together for up to 8 years based on this long-term data set. And so here you can see how their home ranges exclusively overlap with other pairs. The male home ranges are a little bit larger because they're incredibly territorial, and males patrol boundaries a bit more. And they're also the only venomous primate, and their venom is principally used to guard these territories. They also engage in social foraging. This is especially for gum, where you'll see 2 to 4 animals in the same tree. And we also have evidence that young animals, young slow lorises, which don't disperse up to 3 years, it can be as early as 18 months, but it's sometimes up to 3 years, they actually learn to eat this food from watching siblings and their parents. They only give birth to 1 offspring per year after 6 months gestation, and they aren't seasonal breeders. They can have their offspring any time of the year. We do know from some wild studies and from captivity that slow lorises emit a range of sonic calls. These can be in territorial situations where they're whistling at a neighbor, threatening situations, for example, when they're caught to be measured, or during social contact when they're playing with each other or they are associated with young ones. And when we tried to first record these calls in a captive situation in a rescue center, we noted that all of these calls had ultrasonic components. Although they'd been studied in captivity in zoos a bit, I think people didn't realize they would have such an incredible range of ultrasonic components. And what we were finding is our microphone at that time, which was limited to 16 kilohertz, just cut off all the calls. And and so that led us to think potentially there were some ultrasonic calls. In addition, Elke Zimmerman, who did a lot of work on slow lorises, the Bengal slow loris in particular, in the 1980s in captivity, she suggested that juvenile slow lorises emit a pure ultrasonic call in distress contexts and also in novel settings of moving into a new cage. But until now, until our work, there had been no evidence from the wilds. You might have already seen this video as a teaser to this talk, but I think it's nice to show it to you again, just so you can see the calls. Hello, my name is Professor Anna Nekaris, and I'm the director of the Little Fireface Project. One topic we don't talk about very often in regards to slow lorises are their vocalizations. Many people think they're incredibly quiet and incredibly solitary. In fact, they're incredibly social and they make many different calls. And in this video, we're going to introduce you to the main ones. One of the main calls made by slow lorises is the whistle. They do this during the breeding season or breeding time. They do this to attract mates, and they also do it in a territorial context. A second call made by slow lorises is a pant or pant growl. And this can sound a little bit like a stuck vacuum cleaner, and they make it when they're upset or when they're fighting. Another sound made by the slow loris in aggressive context is a chitter. This is when they're also agitated or annoyed, and a chitter can also be made by a parent to its offspring who's being particularly bothersome. Lorises also make some calls when they're very, very upset. If they're being caught or harassed by a predator, generally in my experience, it's a human, they may squeal, or they even may make an alarm call that is very, very sad and very terrifying. And I've even heard this sound when a loris was being electrocuted on a live wire, which happens sadly very frequently. Infant lorises also make a unique call called a zik call, and they do this with a contact to their mother, who may make a click call in reply. And there's another unique call made by the slow loris, which we have called the crow call. That sounds just like a crow cawing, and until now it's only been heard in the Sumatran loris, Nycticebus hillary. A final call that is really exciting that we have identified in the Javan slow loris is one that you cannot hear yourself as a human. It is pure ultrasound at 46 kilohertz, and we have called it the doublet click. Javan slow lorises live in family groups of up to 2 parents and 4 offspring, and this group can make this call hundreds of times per night, up to 200 meters away, and they use it to keep in contact with other family members. So I hope this video has allowed you to see that lorises are not so solitary after all. There we go. So those are the calls. And I gave you the teaser to the doublet click. So yes, we did find that they make a pure ultrasonic call. And the purpose of today's talk is to define the acoustic and temporal structure of that call, to examine what its behavioral context is, or show you the data of the behavioral context, and to consider its potential function relating to 4 factors that mammals often use with ultrasound, including echolocation, which in fact is used by tarsiers, resource advertisement, social group cohesion, or an anti-predator strategy. So we conducted this research in West Java near a village called Cipaganti in an agricultural agroforest environment, which you can see here. We are on the side of an active volcano, Gunung Papandayan, and the actual ultrasonic data were collected from December 2015 to May 2016 from 14 known individuals. And this was done by my student, Dan Gira, who literally went out every night with the recording equipment, just trying to see if these guys made an ultrasonic call or not, pointing the recording equipment at animals, lying in a hammock all night, holding the recording equipment at animals until we discovered the call. It was very evident it was coming from the lorises. We confirmed it was coming from the lorises. And that was about the first 6 months of the work, from June to December, and then December to May 2016, working on recording as many as possible. Whenever the doublet click was heard, we did all occurrences sampling to record the associated behavior, be that affiliative, aggressive, foraging, feeding, or resting. Foraging is looking around for food, kind of a traveling behavior, whereas feeding is actually feeding on the food. Then I also include here 2 years of data from January 2020 to November 2021, using instantaneous sampling, where we record behavior of lorises every 5 minutes, and the vocalizations heard during that time, the audible vocalizations. In order to record these calls, we were using an Echometer 3 Plus and an SMX UT ultrasonic external microphone. It was a directional horn, so pointing right at the animals and trying to isolate external sound. Here you could see we went up to 256 kilohertz because we didn't know initially what the range of the sound would be, and we didn't want to cut off the sound. And then we used real-time expansion to analyze the calls. And later on, we actually have switched now to the Echometer Touch 2 to monitor ultrasonic calls after we identified them. And so the team goes out regularly now with an iPad with that Touch 2 to continue looking at the calls. Moving on to the results. So we detected a single type of pure ultrasonic call, and we analyzed 791 of those. Of course, sometimes you're trying to record this call, you cut off the beginning, particularly. So we were able to isolate 791 that we could analyze in 552 observation hours. We found the maximum propagation distance we could record or detect the call was at 131 meters, but we feel it probably does go up to 200 meters, looking at the size of our fields and how animals communicate with each other. So of course, again, we're working in an agricultural area where there may be no trees in between one hedgerow and another. And that's possibly why this call can go so far. It's emitted purely in the ultrasonic frequency range, and that is at about 46 kilohertz. But you can see that the bandwidth limits mean it can go all the way up to 62 kilohertz, which is pretty rare for a primate. Both sexes and all age groups emit this call. But when I put 4 individuals at once, we have seen it used as a kind of counter call in family groups. So that's 4 individuals counter calling to each other in a single bout or a single night, a single recording period. It is emitted most often during affiliative social behavior. So of 532 calls where we could record the associated behavior, 359 or 68% of the time it was during social behavior, and 16% of the time it was foraging. So that doesn't mean that it wasn't social foraging, because of course these guys are only 1 kilo. They're very small, they're high up in trees, it's dark at night. And although we know that our focal individual was foraging, it doesn't mean that there wasn't another animal nearby. You can see some of the variables in that corner. I think I need to make myself small for you to see the juveniles, that we had calls from adults, subadults, and juveniles. And they're very similar to each other. The call duration is somewhere between 9 and 10 seconds. The dominant frequency is around 45 kilohertz. And you can see some other interesting features about the duration of the clicks, the inter-doublet click duration, the amplitude, and some small differences. For example, the frequency range is a bit higher in juveniles. The maximum frequency is a bit higher in juveniles. And the number of clicks per call is a bit higher in subadults. None of those are statistically significant, however. Let's see if I can move forward. There we go. And here you can see the composition of the call. You've already seen this in the in that video as well, but it is composed of 1 to 2,141 paired pulses. And this is what we call the doublet click. So you could see these little pulses. And the single bout generally consisted of about 10 calls. The longest bout we heard was when these 4 individuals in a single social group called to each other all night. So a lot of the data that we had comes from that bout. And we had an average of 6.66 bouts per active period. So I have to say, we didn't record all of those. This was we sampled some of those, but we have many, many nights where these guys made this call in the 6 months. We used discriminant function analysis to see if we could tell which age and sex was making the call, and we could. So the model statistically correctly classified the age class and even more so the sex class. And so potentially we can distinguish males and females, and potentially the lorises can distinguish each other. When we look at the other vocalizations we recorded in 2020 and 2021, you could see that the audible vocalizations are far less frequent than these ultrasonic ones. In a whole year, in nearly 40,000 and 25,000 sample points, we recorded audible calls only 90 and 134 times. These were mainly whistles with some other calls such as chitters, growls, and screams. I must say, the chitters don't go very far in the forest, and you do hear them quite often in captivity. So it could be something about how far they travel that we didn't record them so much, but they certainly don't occur very often in the wild. So what is this call for? Well, we found no evidence for echolocation or resource advertisement in the doublet click call. I mean, potentially when those animals are foraging, they could be resource advertising, but in general, like I say, it's likely there was another animal around when they were feeding, because that's what happens most of the time. For social group cohesion, however, we really feel that's the purpose of this call, because most of the time they were calling to each other where we at night could visibly see two animals making the call to each other. This is common among birds and primates to maintain cohesion with conspecifics out of visual range. And in the past, I used to see one loris magically come to another, even from hundreds of meters away. And I always wondered how they knew exactly where that other loris was. And it's probably they were making ultrasonic calls. An effective contact call should be encoded with information about the caller's identity and the distance, and we could identify age, class, and sex. We know that actually a loris can move up to about 6 kilometers per night, but when they're together, they are often within about 100 or 200 meters of each other when we're doing our radio tracking. So this is a good distance to call to each other. They also park their juveniles and infants. And so it's a good way to keep contact with those parked, potentially vulnerable youngsters by having this click call. It also could potentially help to reduce extra pair copulation, strengthen the pair bonds of these unimale, unifemale animal social organization, and maybe reduce encounters with aggressive extra-group conspecifics. Because if you could call to each other and stay away, having a venom that's used in interspecific competition, it's very important that you can avoid conflicts which can be deadly. It also may facilitate predator avoidance. Here you see an Indian palm civet. We have a few civets at our site. We have leopards, but we've never seen a loris killed by one of these, only by domestic dogs. The ultrasonic calls are only used during the active period. Lorises in general are cryptic with silent movement. They have camouflage. They have a seasonally changing colored dorsal stripe that we propose facilitates background matching. And so this doublet click call may have advertised not only to communicate with each other, but to avoid advertising to predators. And also allowing communication with group members while remaining inaudible to predators can help maintain social cohesion and reducing energy demand. And finally, I think what's really important about this call, lorises are considered slow, solitary, and rather boring. And now we know they are not only incredibly social, living in these complex groups, but they have this unique call that helps to facilitate these complex social behaviors. So I'd just like to thank you for listening and direct you to our website if you'd like more information, or we also have a YouTube channel where you can see this and many other videos of the behavior of the slow loris. We're on Instagram and Twitter, and you can follow us on Facebook as well, or visit our Etsy shop where you can help support our project. And I'd also like to thank the funders who have been supporting us, including through the pandemic, because we've been able to continue this long-term 10-year study despite the pandemic. And I really want to thank everyone who's been supporting us. I will be happy to answer any questions.
Thank you so much, Anna. we can't hear them, but I'm assuming people are doing this all over the planet. There you go. It was a really wonderful talk. You've set Twitter alight with like lots of facts about slow loris. So we're going to have to look at those a lot later on. But we've got questions kind of like building up already. So, and I've got a few as well, just to sort of add things in. So first question is, why do you think slow loris evolved to use ultrasound, but not other groups of primates?
It's a very good question. They have a very good hearing as well. It's something I didn't mention in the talk. So they have the ability to hear ultrasound, but they've been evolving for a long time. So they separated from other primates at least 23 million years ago, and probably a bit earlier. So with all of their other incredibly unusual adaptations, like being venomous, like being slow and not leaping, they're the only true non-leaping primates. Possibly this anti-predator strategy would have been the original function, to be as quiet as possible along with all of their other quiet features, which is something you're lacking in other primates. Wow.
You're getting lots of compliments. The chat's going crazy as well. And you've got a question
here. Thank you, everyone.
We start fantastic talk. How did you measure accurately detection distances to calling lorises? Was it by subsequent location of the individuals?
Yes, so we had the individual in view, and then we had the microphone pointed at the individual, and we just walked until we couldn't detect the sound anymore. So it may not have been incredibly accurate, but there's also a point where you can't really walk backward anymore because you're on the side of a mountain. Yeah. So that's why we also suspected it can go a bit further than 131 meters. But that was the safest backward distance we could walk from an individual that we could still see.
Yeah, walking backwards at night with shear drops is probably not recommended. Just don't try this or try it at home. Just don't try it outside the home. Another quick question. Really interesting talk. Thank you. As you can identify age and sex from these calls, are there any plans to use these vocalizations for conservation monitoring? Additionally, can you identify specific individuals or group members?
That's something we have a student working on now. So we've been continuing, as I said, to collect the calls, and those data remain to be analyzed. So there's a student interested in that topic. And we're hoping we can identify individuals. So that will be very interesting. Certainly from the sex perspective and the age perspective, we have been looking at using those passive acoustic monitors and to see if we can see in forest patches around the district where we're working. First of all, if lorises are present there, but we know, because we know their general home range size and their general group size, you could at least predict in one area in a very territorial species, if you have a male, if you have a female, if you have some youngsters. So that would be quite a good way to tell, to start getting some population estimates from that perspective. It's just a matter of going through all those data to find the calls within a passive acoustic monitor and have some sort of training to detect them without having to do it all manually.
I've got a sneaky question. Being host, it means I can ask one. As you might well know, I'm into bats, and I noticed that a lot of your parameters that you were measuring when you were measuring the ultrasonic calls, were you using kind of the parameters that we kind of use in the bat world as well? Because it looked very similar. Or did you kind of just come up with those naturally, or how did the
yes, largely we started with bats, and we also looked at the tarsier and the mouse lemur studies that had detected ultrasonic calls in mouse lemurs and tarsiers, and so I think because ultrasound is relatively rare in primates but is very common in bats, that was why we chose those parameters. No, and we also wanted to make sure we weren't getting bats, because we have quite a few bat species in our area, and we wanted to make sure we were recording lorises and not bats, because I know that in some other nocturnal primate studies there had been confusion with bat calls and primate calls, and but we were able to isolate bats. Excellent. And I think you confirmed that as well, because Paul has heard the call. And you know how to make it audible, and I have to learn.
We first met up in Malaysia, and you brought some calls to me, and we sat with a beer after the conference, and a big group of your researchers, and we all played back slow loris calls, which is, that's how we rock and roll when we get a right, back to proper questions rather than what I'm asking. A question from Twitter. I find it interesting that juveniles' range is higher, like humans. Curious as to the length of vocal folds, or is it something else?
Oh, that would be very interesting to know. We're actually going to start doing some anatomical dissections of lorises for another kind of study about facial musculature. So that could be something that we could do at the same time as have a look at their vocal cords. They are different in other ways. I mean, they're obviously much smaller than the adults. So there could just be something about development of the sounds that they make. But they're more vicious as well, and they are more aggressive than adults. So maybe that would also be interesting to know if the sound they make is slightly more threatening to other lorises, because they could potentially be more vulnerable if they weren't so venomous when they're parts. But yeah, but it's something we could definitely pursue, because we're going to start very soon a dissection study. So we could add that to the list.
Excellent. Hi, Anna, great presentation. Do you think that ultrasound is related to the small size group because the need to communicate close, like in porpoises? So so the comparative study of porpoise communication and slow loris communication.
That could be possible as well. Of course, in most nocturnal primates, even when they're semi-gregarious, they're usually unimale, unifemale, or something. Maybe one male and 2 or 3 females. So they tend to, animals at night tend to have small groups. So it could make sense. Yeah, this sort of call. Yeah, I suppose so. I mean, it's a good way to communicate, because it doesn't transfer very far. It's a good way to communicate with a smaller number of animals. So if you're a bush baby, well, actually, though, bush babies are nocturnal and have incredibly loud calls, but they also are very fast and can escape predators quickly. So I think it's a combination of small group size, but also vulnerability due to their slow locomotion. So they want to announce themselves to their group members, but they don't want to be noticed by predators.
Well, in actual fact, the next question is perfect, because do the predators of slow loris hear ultrasound? So
the main predators are snakes, pythons. So I would say no for them. And we have some serpent eagles, but they're dawn and dusk hunters when the lorises aren't making that call. And they're monitor lizards. So probably not really. And it also sounds very much like insects. It's really camouflaged within the insect and the bat calls. So I think it's a confusing call as well for predators. Can confound the predator, I guess.
God, there's just questions flooding in. I hope you're okay. You could be here for hours. Great talk. Can you talk a little more about the function of those calls in pair bonding and how it would reduce extra pair copulations?
Yeah, so the adult males and females they frequently call to each other, and we consider actual gregarious social behavior when the animals are in about 5 meters from each other. And again, when you're out at night, it's difficult to always even see the animals at all. So when you see them together, they're together about 10 to 20% of the time where they're physically together, and or within this 5 meter boundary. So they have the potential to get together very, very quickly. And they're very territorial, where a male and a female overlap each other and not other adults. We do have extra pair copulations, however. So we say they're unimale, unifemale, but we don't use the word monogamous, because we know that other males come in and try to mate with that female. So males do other unique things in the group. They care for the offspring, they carry the offspring, they play with the offspring, they contact the offspring. So I think if you have this regular communication between the adults all year round, even that 1 or 2 weeks a year the female's in estrus, she still may choose her male over these other random guys that they can come in from several home ranges away to try to get a chance with that female. What we don't know, however, is who the daddy actually is, because we've never had permission to do genetic analysis of paternity, which is something else we've just got permission to do. So that mystery will be answered in the next year or two. And it will be fascinating to know if the father, this social father who's intensively caring for offspring, who's often very romantic with his mates, who hangs out with her and calls all night long with her, if that actually means he has a better chance of paternity. That will be the really interesting thing. We, however, have this one guy called Fernando, and we think he might be the father of everyone. So then that answer will be very confusing. So maybe it's a way to try to convince the female, if I call to you every night and I'm really nice to you, will you please let me have your offspring?
So again, so I love this idea of a group of animals I know so little about, because the questions are completely different from what I'm used to. But do captive specimens have the same range of sound? So is it learned behavior?
Well, the interesting thing in captivity, because they're very territorial, they often kill each other, or they have infanticide of infants because they're kept in very small cages and their home range in the wild. In our scenario, in an agroforest, which is really rich in food, is about 10 hectares. But if you get into a forest environment where they have to travel further for food resources, they could have a home range of up to 30 hectares, which is very large for a 1 kilo animal. And so you can imagine in a zoo setting where they have a 3 meter by 3 meter cage, often they're kept alone. So they're often kept solitarily. Sometimes they don't even have the correct species together in captivity, and they're relatively rare in captivity. But what I can say is, from the rescue center situation, the call we hear the most often is the whistle, and they tend not, and the chitter as well. They do it a lot in captivity. Again, those are aggressive and contact calls or territorial calls. So we don't hear that ultrasonic call very often in captivity. Almost not at all. I think we recorded it once or twice, and we tried with 2 different species to record that call in captivity, which makes me think that that contact of being 100 meters apart or 50 meters apart is when the call is used more than when you're next to each other, when you start using audible vocalizations.
So yeah, I think you may have answered this in part, but it's somebody else that's doing something similar on tarsiers in the Philippines. Can the pure ultrasonic call of loris distinguish between species or even subspecies? I am looking for similarities in Philippine tarsier calls, and if their calls can be used to uncover a cryptic species complex.
It would be great to know that answer. So we did try to record pygmy loris and Bengal loris in a rescue center in Vietnam to see if there were differences. But like I said, we couldn't actually record an ultrasonic call in captivity. And there's not been another study like this in the wild yet. So if anyone has data from any other loris species, it would be great to know. But we do have a study starting of Bengal slow loris in Bangladesh, where we can potentially start recording ultrasonic calls. And that would be a really good opportunity, because the Bengal slow loris is genetically most closely related to the Javan. They're about 2 million years apart. So if the most closely related ones can be different, then it would be a really great news for potentially uncovering others. So with tarsiers, there's a lot more data. So it would be interesting to see with tarsiers if we could distinguish the ultrasonic calls by a species.
I've got 3 more questions. I'm determined to get through all 3. So how many lorises have you surveyed, and what do you think the total population is?
Oh gosh, as in, do you mean on Java? I guess that question probably means. We have done island-wide surveys of Java. Interestingly, we found incredibly low numbers, nothing, as in no animals, to very low densities in national parks, which are much higher altitude, and our secondary growth forest, which doesn't necessarily have gum trees, which are the main food of lorises. So they do better in non-protected agroforest, which is heavily disturbed, where they're more likely to be hunted and where they're more vulnerable to human change. So if you're doing IUCN Red List categorization, you don't include that area outside the protected areas. So we don't have a total population number, but we do know that their numbers are still dramatically declining, because there still is trade, there's still habitat loss, and trade for different reasons is increasing. But in our area, we probably have about 200 animals where we're working.
So the last 2 questions. One, you probably know on your next study, but is there an extra organ for making this ultrasonic noise?
Yeah, I don't know. I have no idea. It would be, again, that could be something we pursue when we start doing the dissections. So when people have looked at dissections of lorises before, they it wasn't known, so I don't think they pursued looking for that, but they have checked the hearing, and their hearing is very good, and their hearing perceives ultrasound. We know there's been some really nice hearing work of them. So ears might be important as well.
Excellent. And the last question, what was the analysis process like? Did you use classifiers, or was it more of the analysis, manual, as in with software and things like that? And did you clean the recordings up, remove background noise? Did you process the data in specific ways?
Oh, yeah, that was about a year of analysis actually, and it was a huge amount of cleaning the background noise. It was all manual. There wasn't, it was literally going through hours and hours of data, manually selecting those calls. And then, and that was with Raven, we used Raven. And then once we were able to get all of the parameters of the call, that's when we did the discriminant function analyses.
Brilliant. Thank you so much. Well, if you're willing, there are still more questions appearing in the Q&A. If you're willing to answer a few more, then please do, because obviously you've set our audience alight with that talk to start off with. So thank you so much for that talk, and hopefully you'll stick around, and we'll proceed with the next bit. Thanks again, Anna.
Good luck to the other speakers. Thanks for such a wonderful audience.